As we know that some hair-like structures are present on the surface of the leaf or plant, even entire body of the plant. These are called as trichomes. So I'll show you today how we can observe these hair-like structures under the microscope and how beautiful structures these are. So we have taken one peel of the leaf of another plant, and the peel is under the treatment of toluidine blue, that is one stain, blue color stain. And this here in the stain we will keep it for at least 2 to 3 minutes, so that the peel with the trichomes hair-like structures on the surface of the plant body can be stained, and these will observe under the microscope. So the sample is treated with the toluidine blue stain, and the extra stain is washed away with help of distilled water. So next sample we have to mount on a drop of water only. Extra piece of leaf we have to remove, because the trichomes are present on the surface of epidermis. So only useful part we are keeping with us on the slide. Waste material removed from the specimen. This has to be covered with the glass cover, that is also called as cover slip. Carefully, without formation of air bubbles, we have to spread over it by use of needle. Yeah, and the extra water can be absorbed by use of tissue paper. So now slide is on the stage of microscope, and we will observe how trichomes look like in this particular plant, and the plant name is Coleus foscoli. Right, by use of a smartphone camera we will visualize. This is at low power, so here we cannot see that much things as we call is wonderful, right? With high power we will observe the presence of, here also you can see these pores, round structures are stomata only. So at higher resolution we will observe better and clear trichome. The leaf. Small trichomes. Protect.